Hi everyone, welcome to my channel. Today I'm going to talk about dental plaque, its composition, its formation, and different plaque hypotheses. So first, I want to talk about supragingival plaque and subgingival plaque. So this is supragingival plaque, and this is subgingival. So first, I want to tell what is the difference between both. In supragingival plaque, you can see aerobic bacteria, okay? Because here oxygen is present. As you go deeper, the type of bacteria you see is anaerobic bacteria, okay? And it does not have oxygen-rich environment present. And the type of bacteria is gram-positive, and it changes to gram-negative over here, okay? Now, plaque has both organic and inorganic components. In organic components, it has polysaccharides, proteins, glycoproteins, lipids, whereas in inorganic, it has calcium, phosphorus, sodium, potassium, and fluoride. Okay, so supragingival components are derived from saliva, whereas subgingival components are derived from gingival crevicular fluid. Okay. So in this slide, we have talked about supragingival plaque, where there is aerobic bacteria and there's gram-positive bacteria. And as you go deeper, there's anaerobic bacteria and you see gram-negative bacteria. Composition, you see organic components and inorganic. Organic, there is polysaccharides, proteins, glycoproteins, lipids. Inorganic, you see calcium, phosphorus, sodium, potassium, fluoride. Okay. Next, before we talk about different complexes, we will first talk about plaque formation. Okay. Now, the first step in plaque formation is pellicle formation. Within seconds, the tooth gets covered with pellicle. Okay. And this pellicle consists of, so this happens just within few seconds. It consists of glycoproteins and proline-rich components and the other molecules that serve as attachment to bacteria. So bacteria gets attached to pellicle. Okay, so pellicle formation happens within seconds. Next is adhesion and attachment of bacteria. Okay, adhesion and attachment of bacteria happens within minutes. Pellicle formation happens within seconds. Okay, so what happens here exactly? So suppose this is the pellicle formation. This is happening within seconds. Okay, now bacteria gets attached to this. This attachment is called as initial adhesion, okay, or primary adhesion. So what happens in this adhesion is, this is where bacteria attaches on pellicle. Okay, now there is firm attachment above that. Okay, so this firm attachment is nothing but secondary adhesion that happens between bacterial molecules, that is bacterial adhesin molecules, and the host pellicle receptors. So host also gives a response, right? So there is a reaction between bacterial adhesin molecules and host pellicle receptors. Okay. Now, so pellicle formation occurs within seconds, and then adhesion and attachment of bacteria. First, on the pellicle, primary adhesion happens, that is, bacteria attaches on the pellicle, and about that secondary adhesion happens, where there is interaction between bacterial adhesion molecules and host pellicle receptors. Okay, now the primary adhesion here, we can see what kind of forces is. We see weak, reversible van der Waals forces and electrostatic forces. Okay, and here the secondary adhesion, it is irreversible strong interactions. All right. Now comes colonization and plaque maturation. This happens within 24 to 48 hours. So pellicle formation occurs within seconds, adhesion and attachment of bacteria within minutes, colonization and plaque maturation happens within 24 to 48 hours. Now, first we saw pellicle, then we saw initial attachment, then we saw firm attachment. Okay. Now, what happens here is there is initial colonizers. Okay. They are usually green complex bacteria, which I'll tell about what are the different complexes: green, purple, yellow, orange, and red. We'll talk about it in the next slide. But here, what happens exactly is there is, so let me just draw a diagram just so that, so first layer formed was pellicle, which happens within seconds, and within minutes, there is attachment of bacteria. Okay. Now what happens? There is primary adhesion, that is, first it gets attached, which is weak forces, and then secondary adhesion where it gets strongly attached and irreversible attachment happens. Now colonization and plaque maturation happens within 24 to 48 hours, where there is this specific initial colonizers provides receptors for other bacteria. So what is happening here? Initial colonizers are provides receptors for other bacteria. Okay. Here is when the type of bacteria environment changes from gram-positive aerobes to gram-negative anaerobes. Okay. So this is about plaque formation. So first you see pellicle formation, which happens within seconds, and adhesion and attachment of bacteria happens within minutes. Primary adhesion and secondary adhesion can be seen. Primary adhesion, what happens? Bacteria attaches to the pellicle, and the type of forces here is weak van der Waals forces and electrostatic forces. And the secondary adhesion has irreversible strong interactions. Colonization and plaque maturations occurs within 24 to 48 hours, where the primary colonizers provides receptors for other bacteria, and type of bacteria over here changes from gram-positive aerobes to gram-negative anaerobes. Okay. Now we are going to talk about biofilm. So what is biofilm? So let me first, before I explain what is biofilm, let me show different kinds of complexes. So this is yellow complex, this is red complex, this is orange, blue. So what happens? First there is, if you imagine, this is the pellicle, okay? And initial colonizers are green complex, okay? First they will attach, and then as you go higher, this is going to be an anaerobic environment, okay? The main complex to remember is red complex and orange complex. Okay. P.gingival is b.forsythia, t.denticola. All right. Now, what is biofilm? So we understood that in our oral environment, oral biology, there is a lot of different kinds of bacteria that is attaching to pellicle. First, it's going to be initial colonizers, and then they're going to provide receptors for other bacteria. So what happens exactly here while this plaque is forming? Now here plaque has formed and it has matured. Right. There are fluid channels that you can observe. Okay. This is plaque mass over here, and you see different kinds of bacteria. Okay. So in the plaque mass, you see fluid channels run through plaque mass, permits the nutrients passage. So what are these fluid channels mainly for? So that there is exchange of nutrients. Okay. Now, this bacteria over here can communicate with this bacteria to encourage the growth of beneficial species and discourage the growth of competing species. Okay. So what is happening over here? This bacteria is going to communicate with this bacteria. Okay. And what does it do? It encourages the growth of beneficial bacteria and discourages growth of competing species. Okay, that is nothing but quorum sensing. All right. So we talked about biofilm, that is fluid channels run through plaque mass, permitting the passage of nutrients. And quorum sensing is nothing but how one bacteria communicates with other, and it encourages the growth for good bacteria, beneficial bacteria, and it will make sure that competing species are not growing. All right. Now coming to plaque hypothesis. Okay. So let me write it in a new slide. Plaque hypothesis. There is something called as non-specific plaque hypothesis. Here, they said that as long as there is more amount of plaque, there is more amount of disease, which is not true. Next came specific plaque hypothesis, which is a little bit true. That is, there is something specific bugs in our oral environment and it's causing disease. But they did not mention anything about how the host is maintaining their oral hygiene, how their diet. So it is also not true. Now, coming to the third hypothesis. Third hypothesis is ecological plaque hypothesis. Okay, here they believe that mouth is an ecosystem and bacteria, host factors such as smoking, diabetes, everything contributes to plaque. And this is true because just because there is bacteria, you don't get plaque. If you do not maintain good oral hygiene, if there's a lot of smoking habits, a lot of diabetes, then that will cause more and more amount of disease. Okay, so plaque is nothing but it's like a sticky white film that coats teeth and it has bacteria. And we studied how much time it takes to get there. Okay. Now, in this, the important things to remember is red complex, orange complex, but just try to, if possible, remember actinomyces species. It can be seen in healthy gingiva and it can also be seen in root caries. Okay. And let me just make sure that I underline the important species. So that's what I wanted to tell. Okay, so just for a quick recap, we studied about supragingival plaque, subgingival plaque. Supragingival plaque is about gingiva, and what happens over there exactly is there is a lot of aerobic bacteria, gram-positive bacteria, and it gets its nutrients from saliva. Subgingival gets from gingival crevicular fluid, and it has anaerobic bacteria, mostly gram-negative. And most important thing to remember is red complex and orange complex. Also remember P. gingivalis can be seen in chronic periodontitis. Okay, these are severe conditions. That's why red complex is important. p.denticola can be seen in NAC, and actinomycetic comitants can be seen in aggressive periodontitis. Okay. So next, coming to plaque formation. So pellicle formations happens within seconds, and you can see here for the pellicle, it forms within seconds, and then you see attachment of bacteria to the pellicle. That is adhesion and attachment of bacteria. Primary adhesion has weak van der Waals forces and weak electrostatic forces, whereas secondary adhesion has actually more stronger interactions, which are irreversible. Colonization and plaque maturation, 24 to 48 hours. Here what happens is primary colonizers provides receptors for other bacteria. Here you can see a shift from bacteria that is from gram-positive aerobes to gram-negative anaerobes. Okay, biofilm. What happens in the biofilm? Basically, you see fluid channels where nutrients are flowing in the plaque mass, and also there is communication happening between bacteria so that the beneficial bacteria are supported and competing species are removed, and that is called as quorum sensing. Okay, and next, plaque hypothesis. Non-specific plaque hypothesis and specific are not true, because non-specific says that there's more amount of plaque, more amount of disease. Specific says that there's some specific bug that causes disease, and ecological is marked as like an ecosystem. Bacteria is there. Host factors are smoking and diabetes. Okay, so that's all for today. Thanks for watching.